9.58 seconds. That is the fastest a human being has ever moved across 100 meters of flat ground under their own biological power. Not 9.6, not approximately 9.6, 9.58. At peak velocity during that race, Usain Bolt's body was traveling at 12.4 meters per second. That is 44 kilometers per hour, on two legs, with no engine, no wheels, and no external energy source whatsoever. And here is the detail that should stop you cold. His foot was in contact with the ground for just 0.086 seconds per stride. In less than 1/10 of a second, his body had to absorb hundreds of kilograms of impact force, redirected, and converted into forward propulsion. Every single stride, 41 times in a row. Now, every sports broadcaster, every coach, every highlight reel will tell you the same thing. Bolt was fast because he was powerful, because he trained harder, because his legs produced more force than anyone else's. And that explanation sounds right. It feels complete. It has the satisfying logic of effort equals result. But here is the problem with that explanation. Researchers at Southern Methodist University, led by biomechanist Peter Weyand, measured the actual ground force outputs of elite sprinters and found something that directly contradicts the popular narrative. Bolt did not produce dramatically more muscular force than other elite sprinters. His muscles were not the primary differentiator. So if raw muscular power doesn't fully explain 9.58 seconds, what does? What we are going to examine in this video is a biological mechanism so precisely engineered that material scientists have spent decades trying to replicate it artificially, and have only partially succeeded. It is called the stretch shortening cycle, and it operates through a tissue in your body that most people treat as an afterthought. In 2013, researchers at the University of Queensland published findings in the Journal of Experimental Biology showing that the elastic energy return capacity of the human Achilles tendon can reduce the metabolic cost of running by up to 35%. 35%. That is not a marginal efficiency gain. That is the difference between a biological system and a mechanical one. And in Usain Bolt's case, the architecture of that system was operating at a level that the science of biomechanics is still working to fully quantify. To understand what Bolt's tendons were actually doing in Berlin in 2009, we need to start at the molecular level. A tendon is not a rope. That is the first and most important correction to make. The popular mental model, tendon as passive connector between muscle and bone, is not just incomplete, it is functionally wrong in a way that obscures the entire mechanism. A tendon is composed primarily of type 1 collagen, a protein arranged in a triple helix molecular structure. These triple helixes bundle into fibrils, fibrils bundle into fascicles, and fascicles bundle into the tendon unit itself. Each level of this hierarchy is slightly crimped, arranged in a sinusoidal wave pattern at rest. Think of it like a coiled spring that has been woven, then bundled, then woven again at a larger scale. When a load is applied, when Bolt's foot strikes the track, this hierarchical crimp structure begins to uncrimp sequentially. First, the molecular crimp straightens, then the fibril crimp, then the fascicle crimp. Each level absorbs mechanical energy as it straightens, and then, critically, when the load is removed, that crimp snaps back. The energy that was absorbed during loading is returned during unloading. This is the elastic storage mechanism. And in biological tissues, the efficiency of this return, the ratio of energy returned to energy absorbed, is called resilience. But here is where it gets strange. Human tendon resilience has been measured at approximately 93% in vivo, meaning in a living, functioning system. For context, the carbon fiber used in modern running prosthetics, the blades you see Paralympic sprinters using, achieves resilience values of around 95%. The biological tissue that evolved in your ankle over millions of years is operating within 2 percentage points of the most advanced engineered material humans have ever produced for this specific purpose. That is not a coincidence of numbers. That is convergent optimization. 2 completely different design processes, one biological and one engineering, arriving at nearly identical solutions to the same mechanical problem. Store energy during impact. Return it during propulsion. Lose as little as possible to heat.
Now let's look at how this plays out during an actual sprint stride. When Bolt's foot strikes the ground, his ankle joint undergoes rapid dorsiflexion. The foot bends upward toward the shin. This stretches the Achilles tendon and the plantar fascia simultaneously. Both structures load with elastic potential energy, exactly as a spring would if you compressed it. The gastrocnemius and soleus muscles, the calf complex, are contracting isometrically during this phase. They are not shortening. They are holding tension while the tendon does the mechanical work of energy absorption. Then, as the center of mass passes over the foot and the propulsion phase begins, the ankle plantar flexes, pushes downward, and the tendon recoils. It releases its stored elastic energy back into the system with 93% efficiency. This is the stretch-shortening cycle. And the key insight, the one that changes how you understand sprinting entirely, is this. The energy that powers Bolt's propulsion is not coming exclusively from his muscles contracting in that moment. A significant portion of it was stored mechanically, a fraction of a second earlier, in a passive, elastic structure. Here's what nobody tells you about this mechanism. The muscle, the gastrocnemius, the soleus, is functioning less like an engine and more like a brake and clutch system. Its job during the ground contact phase is to regulate how quickly the tendon unloads. If the muscle released tension completely, the tendon would recoil too fast, and the energy return would be mistimed relative to the stride cycle. So the muscle holds isometric tension. It neither shortens nor lengthens significantly, while the tendon does the spring work. This division of labor between muscle and tendon is called muscle-tendon unit decoupling, and it was first clearly demonstrated by researchers Roberto Fukunaga and Yasuo Kawakami at Waseda University in Japan, using real-time ultrasound imaging in 2001. Their data showed that during walking, the calf muscle fascicles moved less than 3 millimeters during ground contact, while the tendon stretched and recoiled by up to 10 millimeters. The spring was doing the work. The muscle was holding the spring in place. Now scale that mechanism to maximum sprint velocity, to 12.4 meters per second, to 0.086 seconds of ground contact. At those speeds, the Achilles tendon in an elite sprinter is experiencing forces of approximately 9 times body weight during each ground contact. For Bolt, who competed at approximately 95 kilograms, that means a force of roughly 855 kilograms, nearly a metric ton, passing through a tissue roughly the diameter of a thumb. Every stride,
41 times in 9.58 seconds. And the tissue does not fail. Not only does it not fail, it performs this load-store-return cycle with 93% efficiency at every single repetition, in a time frame so compressed that no voluntary muscular contraction could match it. And this next part is what the data actually shows, and it is genuinely counterintuitive. At maximum sprint velocity, the elastic energy return from the Achilles tendon and plantar fascia accounts for a substantial portion of the mechanical work done during each push-off phase. In 2010, biomechanist Art Kuo at the University of Michigan published work estimating that in running, elastic energy storage and return in the lower limb can supply the equivalent of what would otherwise require an additional 35 to 50 watts of muscular power per leg. 35 to 50 watts per leg, per stride, for free. For free, meaning the energy was already in the system from the impact of the previous phase. It was not generated by additional metabolic expenditure. It was recycled. This is the mechanical equivalent of regenerative braking in a hybrid vehicle. Kinetic energy that would otherwise be lost as heat is captured and reinvested into the next propulsion cycle. But here is where Bolt's specific anatomy enters the picture in a way that moves beyond general biomechanics and into individual biological architecture. The stiffness of a tendon, how much force is required to stretch it a given distance, is a critical variable in this system. A tendon that is too compliant stretches too easily. It stores energy, but the recoil timing is too slow for high-speed locomotion. A tendon that is too stiff doesn't stretch enough to store meaningful energy. There is an optimal stiffness range for sprint performance, and that optimal range is determined by the interaction between tendon stiffness, muscle architecture, and stride frequency. Researchers Pekka Komi and Jana Avala at the University of Jyväskylä in Finland have spent decades characterizing this relationship. Their work, published across multiple studies in the Scandinavian Journal of Medicine and Science in Sports, demonstrates that elite sprinters show tendon stiffness values significantly higher than recreational athletes, with the Achilles tendons of sprinters showing stiffness coefficients up to 40% greater than age-matched non-sprinting controls. 40% stiffer. That is not a training adaptation you produce in a single season. That is structural. That is architectural. That is the result of years of specific mechanical loading during the precise developmental windows when tendon tissue is most responsive to adaptation. And, in some cases, particularly in athletes with exceptional genetic predisposition, that stiffness reflects an underlying collagen architecture that was never average to begin with. But this is where it gets strange. Bolt's stride mechanics show another feature that should not be possible given standard biomechanical models. At maximum velocity in Berlin, Bolt's stride length reached 2.77 meters, an extraordinary figure even for his height of 1.96 meters. The conventional model predicts that longer stride length should come at the cost of stride frequency, because longer ground contact times are needed to generate greater push-off forces. Bolt violated that trade-off. His stride frequency at maximum velocity was 4.64 strides per second, while maintaining that 2.77 meter stride length simultaneously. The physics of how a single body achieves both at once, without the ground contact time increasing proportionally, points directly back to the tendon. When the tendon's elastic recoil is powerful enough and fast enough, it can generate substantial propulsive force within a very brief ground contact window. The muscle does not need extended time on the ground to generate push-off force. The tendon provides a large portion of it instantaneously through stored elastic potential energy. The tendon is doing in 0.086 seconds what muscle contraction alone would require significantly more time to accomplish. The question that follows from everything we have just established is the one that matters most from a biological standpoint. How does a tendon become this? And more specifically, how much of what Bolt had was built and how much was inherited? Tendon tissue adapts through a cell type called tenocytes, tendon-specific fibroblasts that synthesize and remodel collagen in response to mechanical loading. When a tendon experiences repeated tensile stress within a specific load range, tenocytes upregulate collagen production and cross-linking enzymes, gradually increasing the tendon's stiffness and load-bearing capacity. This process was characterized in detail by Keith Barr at the University of California, Davis, whose laboratory work on tendon mechanobiology has demonstrated that collagen synthesis in tendon tissue peaks approximately 24 hours after a mechanical loading stimulus and returns to baseline within 72 hours. This gives us a precise biological time frame for tendon adaptation. The stimulus must be repeated with a frequency that keeps collagen synthesis elevated, but with sufficient recovery time to allow cross-linking to complete. Too frequent, the newly synthesized collagen doesn't have time to mature and cross-link before the next load disrupts it. Too infrequent, the synthesis stimulus decays before meaningful structural change accumulates. The optimal loading frequency for tendon adaptation, according to Barr's work, is approximately every 36 to 48 hours, which maps almost exactly to the loading frequency of an athlete in serious sprint training doing 2 to 3 quality sessions per week. Now here's the adaptation timeline that nobody in sports talks about because it is uncomfortable. Muscle tissue adapts relatively quickly. Meaningful hypertrophic changes are measurable within 4 to 8 weeks of a training stimulus. Tendon tissue operates on a fundamentally different biological clock. Meaningful structural changes in collagen architecture require months to years of consistent loading. Research by Henning Langberg at the University of Copenhagen, using microdialysis to directly measure collagen synthesis in the peritendinous space of human Achilles tendons, found that measurable increases in collagen turnover required a minimum of 8 to 12 weeks of consistent loading to produce detectable structural changes, and that full architectural remodeling of a tendon's stiffness profile could take 18 months to several years. Bolt began serious sprint training as an early adolescent in the parish of Trelawney in Jamaica, a region with an extraordinary history of producing elite sprinters, a phenomenon that has attracted genetic, environmental, and biomechanical research in its own right. The tendons he brought to Berlin in 2009 were not built in the months before that race. They were built across a decade or more of specific mechanical loading, starting at a developmental stage when tendon tissue is most responsive to architectural remodeling. And the genetics question must be answered directly. The COL5A1 gene encodes the alpha-1 chain of type 5 collagen, which regulates type 1 collagen fibril diameter during development. Specific polymorphisms of COL5A1 have been associated in multiple studies, including work published by Malcolm Collins at the South African Medical Research Council, with differences in tendon mechanical properties, including stiffness and injury susceptibility. An athlete inheriting favorable COL5A1 variants begins the adaptation process with a different starting architecture. Their collagen fibrils are differently proportioned from the outset, creating a tendon that responds to mechanical loading along a different trajectory than the population average. This does not make the training irrelevant. The genetic architecture is the blueprint. The decades of mechanical loading are the construction. Both are necessary. Neither alone is sufficient. And this is where it stops being about Bolt and starts being about you. Because the biological mechanisms we have described, the stretch-shortening cycle, the tendon's elastic energy return, the collagen adaptation response, are not exclusive to world record holders. They are operating in your lower limb right now. Every time you walk down a corridor, every time you climb a flight of stairs. The question is not whether you have the mechanism. You do. The question is what state that mechanism is currently in and what it is capable of becoming. Let's extract the mechanical principle from everything we've examined and state it clearly. The elastic energy return law of biological locomotion. In any biological system that moves by cyclical limb contact with a surface, metabolic cost is minimized, and mechanical power output is maximized when passive elastic structures, tendons, ligaments, aponeuroses, are optimized to store and return mechanical energy across each contact cycle, shifting the energetic burden away from active muscle contraction and toward passive spring mechanics. This is not a principle specific to human sprinting. It operates across the entire biological kingdom of terrestrial locomotion. The kangaroo is the textbook demonstration. Researchers T.J. Dawson and C.R. Taylor, in a landmark 1973 paper in Nature, showed that kangaroos at high hopping speeds show no increase in metabolic cost per unit distance, a result that is physically impossible if muscles are doing all the mechanical work. The explanation? The kangaroo's massive Achilles tendon, proportionally far larger than any other mammal relative to body mass, stores and returns elastic energy with such efficiency that additional speed costs almost no additional metabolic energy. The animal is effectively bouncing on a biological pogo stick. The horse presents a second demonstration. Equine biomechanics researchers Alan Wilson and colleagues at the Royal Veterinary College, in work published in Nature in 2001, showed that the superficial digital flexor tendon in a galloping horse stores and returns energy equivalent to what 30 additional kilograms of muscle would need to generate if the tendon were replaced with an inelastic material. 30 kilograms of muscle replaced by a single tendon. And in human engineering, the carbon fiber running prosthetic is the conscious attempt to replicate this mechanism artificially. The curved carbon blade is not designed to look like a foot. It is designed to have the same elastic storage and return characteristics as a biological lower limb. The shape, the stiffness, the cross-sectional geometry, all of it is calibrated to achieve the same stretch-shortening cycle that Bolt's Achilles tendon was performing naturally. The fact that it achieves resilience of 95% versus biology's 93%, and that this difference has generated decades of ethical debate in Paralympic sport, tells you everything about how precisely the biological system had already been optimized. The universal law holds across all of these contexts. Elastic energy return is not a performance enhancement. It is the fundamental architecture of efficient biological movement. What differentiates elite performers from the population is not the presence of this mechanism, it is the precision of its calibration.